My name is Dr. Sophie Kerneis, and I am working at Lethbridge College in the Microbial Research Laboratory. And today I would like to present our research on phytobiotics to serve the poultry industry, an alternative to antibiotics. So before we start, I would like to speak about the discovery of antibiotics. The first antibiotic discovered is penicillin. It was discovered in 1928 by Alexander Fleming. And following this discovery, many other antibiotics, such as tetracycline or erythromycin, have been discovered as well. All the antibiotics that are on the market come from fungi and bacteria. No antibiotics that are on the market are coming from plants. So, but who is using antibiotics? Humans and animals. If you take humans, we have been using antibiotics to treat infectious disease and then to prevent infectious disease. In all the minor surgery or all the cancer treatments, you will require to get some antibiotics. In agriculture, we have changed the way we raise animals to feed this growing population. And the antibiotics have been used to prevent infection or to promote the growth of the animals. So today, we have about 80% of the antibiotics that are used for the livestock industry and 20% for the humans. So which is great. Antibiotic is seen as a miracle drug. But we have also a major concern, which is antibiotic resistance. So what is antibiotic resistance? Antibiotic resistant bacteria are bacteria that are not killed anymore by antibiotics. The bacteria they are due to genetic modification. And over time, as we were using antibiotics, we have selected these bacteria that were already present. So today, we have more and more of these antibiotic-resistant bacteria, and the bacteria have a very important genetic plasticity, so they can exchange these genes that are coding for this antibiotic resistance. So the World Health Organization is saying that we need to do something today, because if not, by 2050, we'll have 10 million people that will die of infectious disease that are caused by antibiotic-resistant bacteria. So what can we do? The World Health Organization is saying that we need to prevent infection. And preventing infection is also to find new alternatives to antibiotics, such as phytobiotics for the agriculture. To reduce the use of antibiotics, especially the one that we use for humans purposes. And another way to do it is to develop antibiotics or to identify antibiotics that will be just dedicated to the agriculture. So in my laboratory, we want to identify plants with antibiotic activities, and we have been doing that for many years. We are very localized. We are localized in Alberta, which is great because we have access to this unique ecological zone with plants that have never been tested for their therapeutic properties, and especially not for their antibiotic properties. We are also in an agricultural province, and so we have access to farmers, poultry producers, livestock producers in general. So we have developed a unique plant extract library made of about 190 extracts. We have a biosafety level 2 microbiology lab with a bacterial collection with bacteria that are relevant for the poultry industry. We have the technique established to test the antibiotic properties of the plant, and this is what I will present now. So just a little note on plants. Plants are a source of drugs, as everyone knows. About 50% of the chemotherapeutic agents come from natural products or their derivatives. Cranberries, thyme are used for chicken to prevent certain infection. So we need to find more plants to continue to have this agriculture going and to be able to feed this growing population. So in the Microbial Research Laboratory, we collect plants that are native to Alberta, or plants that are more relevant for the animals that are already part of the animal feed. We collect plants in a sustainable way. We give a plant a number to keep the confidentiality and for the simplicity. And these are places where we collect the native plants. So around Lethbridge, around Okotoks, and in the Crossness Pass. We collect with permits or we collect on private lands. After collecting the plants, the different parts are separated and we dry the plants, we grind the plants, we extract the molecule with solvents such as ethanol or dichloromethane. Then we remove the solvents and we arrive with this dry powder. We put this powder in these little vials with the number. Today we have 190 extract and we are in collaboration. We collaborate with the Natural Product Laboratory at the University of Lethbridge that has about 450 extract. The technique. So the technique we use is called the microdilution technique. The principle of this technique is to put in contact the bacteria that you have here with the plant extract. And if the plant extract has an antibiotic activities, it will stop the growth or reduce the growth of the bacteria. Something that we can see by the cloudiness of the solution. So if the solution is cloudy, the bacteria will be present. If the solution stay clear like here, then it will mean that the plant extract will have an antibiotic activities. We perform this test in these 96 well plates, which allow us to do everything in triplicate and to integrate all the control. So this is a graph that we can generate from the measurement of the optical density that is done every half an hour for 18 hours. So we perform a kinetics. On the y-axis, you will see the optical density at 630 nanometers. And on the x-axis, you can see the time. As you can see on this figure, this is just one example of something that we can find. Here it's with Staphylococcus aureus, which is responsible of food infection, skin infection, but also of bumblefoot on chicken, for example. We can see that we have 4 candidates, and you have the number here, that will do a complete inhibition at 1 milligram per ml for 18 hours. So it will stop completely the growth of the bacteria for this period of time. This extract GP350C will not do a complete inhibition, but will do something that we call a light phase extension. So it will stop the growth of the bacteria for a longer period of time, so about a 10 hours delay, which could be also very interesting. So here, as you can see, we have some candidates. So the graph is just one example. This is a table of some of the extract that we have been tested. We can see here, based on that we have plant extract that belong to different family, and which is in favor of probably having different molecules and different mechanism of action. So over the 19 extract that we have tested for Staph aureus, we have 14 candidates that could be interesting for the agriculture or for the poultry industry. Something that we do in collaboration with Professor Raymond Anderson in UBC Vancouver is that we do a bioactivity directed fractionation. The goal being that from the extract that is active, we will isolate the molecule. We have been able to show that we can do that. For this extract, we have been able to isolate 2 antibiotic molecules. So if you refer to these 14 candidates, maybe we can have more candidates for being the new antibiotics that could serve the poultry industry. Another project that I would like to mention today is the clamping project. So this has been realized by Mrs. Nadia And, who just graduated from the University of Lethbridge and will be conducting her master thesis with me at Lethbridge College. So Nadia has observed that for certain plant extracts, such as the NP856 here, we can see what we call a clumping of the bacteria. So here, if you recognize, the bacteria is cloudy, so very rich in bacteria. And when we put the plant extract, after a very short period of time, it starts after 5 or 10 minutes, we will see a clumping, a removal of the bacteria, of 99% of the bacteria from the solution going into this clump. And this, I think, would be quite interesting for the agriculture to clear the water from bacterial contaminant. So, so this is something to develop further. So in conclusion, I would like to say that we have identified many plant extracts with antibiotic properties against Staphylococcus aureus that I've presented today, but also we have plant extracts that are acting against other bacteria, such as E. coli. We are in the capacity to identify the antibiotic molecules, and we will hope to find more candidates that could serve the human research, but human medicine or the agriculture. And we have identified the clumping activity of plant extracts that will help to prevent the infection and so to prevent the usage of antibiotics as well. What is next? To continue screening, finding more plants with antibiotic activities. The more we have, the better it will be. More antibiotic molecules from these active plants. Two, in collaboration with Dr. Roy Goldstein at the University of Lethbridge, to find their cytotoxic activities on human and chicken cells in culture to avoid that. To continue to test the clumping plants in the purpose of water, to remove the water contamination, such as in E. coli, and to connect with poultry farmers to engage in collaboration. So finally, I would like to thank all the team, all the students that have been involved in this research, and CERC and Lethbridge College that has funded this research, and the collaborators that have playing a very important role in this research. Thank you very much. If you have any questions, you are welcome to contact me by email. And if not, please ask me the questions. Thank you so much.
Great. Thank you very much, Sophie. That was a really interesting presentation. So question has come in. Did you also measure the anti-inflammatory functions of the plant extracts? They might not be antibacterial, but could decrease the inflammation and could be beneficial as antibiotic growth promoter alternatives. I think your mute is on. There you are.
Yes, now I'm unmuted, but I cannot. Okay. So now you can see my face. So thank you so much. No, we have not tested in the lab the anti-inflammatory properties of the plants, but we have, in collaboration with Dr. Roy Goldstein at the University of Lethbridge, we are planning to do that. So for this plant, from this plant library, basically, we are testing different properties. I'm taking in the part of finding antibiotic properties, but Dr. Goldstein is looking for anti-cancer activities for humans, anti-inflammatory, anti-UV, etc. So yes, absolutely, we will be doing that.
Great. So another question is, if you have these products, these natural plant products with antimicrobial activities, are there risks of the microbes developing resistance to these antimicrobial compounds?
Well, everything is possible. The microbes are very smart, in fact. They can develop resistance, or we can, I should say probably more, we can select the one that can resist, because this was even before penicillin was discovered, the penicillin-resistant bacteria were present. So this is absolutely possible, and it's why I said in my presentation that the more we have, the better it will be, because we need to stay, advanced and compared to the bacteria. So we are trying to find a lot of these antibiotic properties in plants, and to find as many candidates as we can to have lots of solutions coming up. And I think with this issue, one of the problems was that the pharmaceutical companies stopped looking for new antibiotics because, probably because of the money issues. But and I think we have to go back before we are in a pandemic. We are stopped. We don't want to have more antibiotic-resistant bacteria coming up.
Mm-hmm. All right. So you mentioned that these were plants I guess, in the wild, if you want to
Yes.
call it that. And so you mentioned that they were sustainably collected, and so let's say that you identify some plant products that are really promising and going through testing in poultry, and it's wildly successful. Would that mean that you'd have to start cultivating these particular plants, or would you be able to collect them in a perfect world, maybe they're weeds and somebody will pay you to come and collect their weeds.
Yeah, absolutely. Dandelion, who has no dandelion in their garden? So yes, we have thought about that, and we have started some collaboration to see when we cultivate the plants, do we keep the same activities or not? And we did. So there's this option to imagine that, as you said, it's very successful. Maybe it can bring a new activities in Alberta to grow this plants, for example, and having fields of the plants that are active. Another also option would be with chemists to try to synthesize the molecules that will be active. And so this is one of the example was with Taxol, which is an anti-cancer drugs. So they first found these activities in trees, and then after that they were able to basically not being dependent on the trees because it takes a long time to get a tree. But so there is still this option, and I'm pretty sure that if it is something very, very successful, everybody will find a way to find enough sources for these molecules.
Okay. All right. Well, that's all the time we have, but thank you very much for your presentation. I really enjoyed it.